Welcome to your first punctibyte for the fall of 2014. For those of you who are new to the whole punctibyte process, we usually like to kick things off with something that is of current events, kind of pertinent information. And I have a couple things I want to just talk to you about really quickly. I've been kind of up on my high horse lately, Freudian slip, regarding the health of your microbiota and the way in which that impacts your overall health. One of the ways in which we're seeing that is in the world of immunology, and your body's ability to mediate a proper immune response depends a great deal on the health of your microbiota. Things like the hygiene hypothesis, right, regarding the idea that when children aren't exposed to any outside antigens when they're young, they don't develop a healthy microbiome, and due to that they are more subject to allergy and asthma. And so a lot of this is developing and evolving, and this was a very recent publication actually regarding the ability of our body to respond effectively to the flu vaccination. And basically this study was looking at mice, and it was looking at the fact that the antibody response in germ-free and antibiotic treated mice was impaired, and was only restored when they were able to restore certain components of the microbiome. So very, very fascinating study, and one that was published in Immunity just this month, and I think I was talking with John Held about this quite a bit. The other one that I want to bring up to you is actually regarding multi-drug resistance and extensively drug-resistant tuberculosis, which is a huge problem right now in South Africa. And this particular article is relating to the link between the negative prognosis of children with HIV and MDR-TB and the role that nutrition plays in that. So certainly a call for trying to help in these developing nations with regard to allowing children to have a greater, a more successful prognosis based upon getting good nutrition. Obviously something worth our time and effort, especially because this week is in fact the acid fast stain, and we'll be actually looking at the fact that Mycobacterium tuberculosis can actually be diagnosed using an acid fast stain. It's one of the only stains that is diagnostic. So I'm actually going to draw your attention to some of the weekly happenings, and we're going to take a look at our calendar. So coming back to our TA site, our group that you can find all of the resources on. I'll be posting the punctibytes under modules as well, and you'll be able to hit on modules and see these punctibytes right here. I'll also try to send emails out to you always to remind you that you can find them here, and then probably even include the YouTube address in my email as well. I wanted to show you the calendar feature one more time, reminding you that on the calendar you can see what labs are going to be happening this week. You can even click on the TA site and you can get, so if you click on microbiology TAs, that will show you which events are pertinent for us, and you'll notice that this week we have lab 3 and lab 4, and that Josh and John are up for open lab, so that just gives you an idea of what's up and coming for the week. You all will be taking over much more this week, and in fact lab 3 and lab 4 lectures will be your responsibility, and so this means you can either come up with a creative way to do the lecture, or you can just practice your lecture delivery skills. And maybe this first time you want to just practice your lecture delivery skills. Though I did want to share with you, I got some informal feedback about doing the flipped kind of strategy where we did for lab 2, and we had them come to lab completely prepared. I asked them just a quick question. This is an informal poll that I took as to whether they preferred that flipped lab, and you'll notice that a majority of those that polled in did prefer the flipped lab. There were some that didn't, but it's really cool to see that we're getting some interesting informal feedback on that strategy, a different way for delivering the lecture. And I know I enjoyed it, helped me feel fresh and new and have a kind of different approach to the day. That being said, let's talk about this week's labs, lab 3 first, and then lab 4. And again, back to our TA group, you'll notice that the TA guidelines are available to you under modules. And in the TA guideline tab, you're going to find TA Lab 3 and TA Lab 4, and those are the guidelines that you're going to want to check out for this week. I'm going to pull over the one for Lab 3 and a couple of highlighted things not to forget, and that is that we will have our first formal assignment that we will assign this Tuesday. You will want to read to them from that gram stain assignment. I would really like to make sure that all the students know exactly what it is that we want them to do, and I really feel like just reading through that assignment with them. You can do that in a creative way, and in fact I would suggest doing it in a creative way, maybe having them respond to you or read through portions of it, and then having them ask questions, but somehow to get that information really out there to them. With those that are giving the lecture for the gram stain, I will likely begin to cover the cell wall structure tomorrow on Monday in class. However, I will not likely finish that. So please be aware that much of the coverage that you will do of this subject is super important to them. So you want to slow down and really take time to tell them what makes a gram-positive cell wall gram-positive. Really iterate the thick layers of peptidoglycan, the presence of teichoic and lipoteichoic acids. What is the difference between the two? Talk to them about lipopolysaccharide, why lipopolysaccharide allows gram-negatives to be toxic, and why why the structure of a gram-negative is different than a gram-positive, including the entire presence of an outer membrane with porin proteins and bronze lipoproteins and whatnot. So use the models that I have, and I am more than glad to get you a number of models to show the 3D kind of perspective of the cell wall structure, or at least to give them a visual depiction of that cell wall structure. Read through these guidelines for more details, but also be aware that this is an important stain. And so if you can designate one of the TAs to be near the sink during this stain, it would be really great to help the students with that artsy step of decolorization, really showing them how long they should decolorize, that if their smears are too thick, that they're probably going to get variable results, and really talk to them about this. I also want to make sure that we clearly differentiate that the acid-fast stain is a completely different procedure than the gram stain. They're not linked in some way to each other, except for in the fact that they are both differential stains. So make it clear that when we're looking for an acid-fast cell, we're looking for the presence of mycolic acid, and particularly for those cells that have a unique kind of acid-fast cell wall. This particular picture is a cartoon of what we would expect to see in an acid-fast cell wall, and you'll notice that the mycolic acid is very distinctive. But also note that the structure of the cell wall is unique. There's some arabinogalactan that is the polymer that is outside of the peptidoglycan, and it's not a stretch to see why most acid-fast cells stain gram-positive, even though their cell wall is in fact very unique, but they do have a very thick layer of polymerous material lying outside of the cytoplasmic membrane. That being said, I'm going to jump into Lab 4 and talk about what we'll be doing on Thursday. Thursday should be less busy with regards to staining, but it is important to start them by doing their capsule stain. So before you even kick anything off with regards to the lecture, have them make a thin smear with their milk culture of Klebsiella pneumoniae. Have them let that sit and dry while we're doing the lecture for this particular lab, because that'll take a little while to happen. The lecture is the only exposure that the students will have to endospores and endospore formation. So please make sure that you lecture accordingly. Let them know how it is that an endospore is built and give some details on that. Allow them to understand the fact that endospores can sustain in the environment for very, very long periods of time. And there is even an article that I've shared with you that is also in the notebook that you can help them understand that it may last for much longer than 100,000 years. In fact, the article that I've shared with you relates endospores taken from 25 to 40 million year old Dominican amber. And so we're seeing a much, much longer period of time, very Jurassic Park-ish in that way. It's also really interesting to relate this to the anthrax scare of post 9/11 and to think a little bit about the different kinds of anthrax. Likewise, botulism has got quite a name because of Botox. So if you want to discuss with them that the botulism toxin is created by an endospore-forming bacterium, namely Clostridium botulinum, that's a really cool kind of current way to relate to something that they might know. We will cover external glycocalyces in lecture, but we do not cover specifically capsules. So spending a little bit of time on what a capsule is, how the capsule aids in increasing virulence of the bacterial pathogen, that would be fantastic. Also take note that Lab 5 will be next Tuesday, so a week from this Tuesday, and that lab is very intense with regards to the calculation of dilution problems. So it's a good idea to have them spend some time with Virtual Edge ahead of time before that lab and to be prepared a little bit outside of lab. That being said, I believe that covers most of what we need for this week. But as is always going to be the case, please feel free to reply to all if you have other things to add and or questions to ask about what will be happening in lab this week. Once again, there is one other thing actually that's really important. Make sure that you recognize that you need to write the pre-lab for Lab 4 and that needs to be accessible for the students prior to Lab 4, so preferably opening it after Lab 3. So if you haven't already written the Lab 4 pre-lab, that's something that really needs to be done. And the other really important thing is that Lab 7, remember we're going to have another flipped lab that day. I've already done the Lab 7 vodcast. So you guys will need to watch that Lab 7 vodcast so that you can write some pre-lab questions for Lab 7 that will be fitting to that vodcast, so that you're encouraging the students to watch that vodcast prior to taking that pre-lab, and that they are accountable for the material in that vodcast. I think that's everything, and I hope you all had a fantastic weekend and that you're looking forward to your first really big week of lab.